Hey,
Hello?
Janet,
Hi, how are you?
sorry about that.
No problem.
Obviously, I need to update my Skype
Yeah,
software. I think.
yeah, I should have maybe made I knew that could be a problem with some of the Skype versions.
Uh-huh.
So,
And
um,
are they also join? Are they going to join us?
so I talked to Celia. She she'll join when and if she's able to. She was on another call.
Okay.
And I haven't heard from Paul, but
Oh,
he should have got the same threads that you did for
okay.
this link.
I
So
think this is much easier. This Google, I never use it, so it seems to just click it, it get in.
yeah, so we use this for some of our other meetings. Like one person organizes it and then just sends the link to everybody, and
Okay, that's
it
easy.
works pretty well for a
Yeah,
multi calls.
yeah, amazing.
Yeah.
Yeah. So should we wait for them or should we just go ahead?
Well, I mean, Paul is the only one. Maybe. I mean, I sent him the same. I sent him an email,
Hmm.
and he should have seen the same Skype, the Skype link that you saw. Let me just check here, make sure everything went through. Yeah, I sent him an email, and then he should see the link also on Skype. So,
Okay.
um, I I don't know. Do you want to wait just maybe 2 more minutes and?
Yeah, yeah. But I'm just kind of looking at you. I
Yes.
still don't structure though. During my lunch, I still trouble to make sense out of those structure. The
Well, like
ribbon,
we
the
can
ribbon
talk
and
about
book
it.
kind of difficult to understand, and I don't have the 3D vision. So
Sure. So, I mean, I sent those so we can kind of start to get a flavor of
uh,
what I did. But of course, there's um, much more data that is not shown. That I couldn't
I
show
can imagine. Yeah,
everything there, but
yeah.
I have all that. We can pull up and look at it in a three-dimensional viewer so that we can rotate it and kind of get a
Okay,
feel for where things are. But
okay.
yeah, this was the first
So
pass that.
I just try to understand when
Mhm.
you do this, right?
Yes.
You download those data from the website, is that right? You download the structure data from the website.
Sure. Yeah. The first thing that I do is I use those PDB ID codes to
Mm.
download those structures, and
And
then
you say you have
prepare
to
them.
prepare those structure for
Yeah.
docking to
Right.
build the what? To build side chain. So you have to do it manually or it's kind of computer program.
I use programs to do that. Okay. Here we go. Just
Or
a
is it?
second here. Um, see who this is. Celia. So, um, is this? Did you call me? Oh, is Paul? Oh, Paul. Hi, Paul. Hi, Paul. No problem. So, iChin is not able to use Skype in a multi-call fashion. So, I did send a Google Hangout link that if you paste into Google our Chrome browser, then iChen and I are both there. It should be in your email. Oh, okay. I think it'll work in Safari.
I use Safari and it worked well.
Okay. Okay. So yeah, he's saying it will work in Safari if you just paste that link, and then we'll all be in the same session. Yeah, or if you want to try it first to make sure it all works, that's fine too. Do I still have you, iChen?
Yeah.
Okay. Okay.
So did Paul try to join it?
Yes, if you put that link in your browser, it should prompt you with whatever you need to join the conversation.
I think I have to download a application called Google Voice, something like that.
Oh, there he is. I see. Okay.
Hey, Paul.
Hey, iChen, how you doing? We'll get rid of this.
Pretty good. How are you?
Good.
Sorry about the trouble because my
Good.
Skype need to be updated.
Yeah, we were just we hadn't really started yet. We were just kind of talking about the preparation of the structures and where that information comes from.
I think
So.
Celia is online. Celia, hi. How are you? Finally. Thanks.
Hi, Celia.
See you.
So I can.
I'm quarantined.
You're quarantined, yeah. He's sick.
Oh no. Well, then Skype is perfect, or Google Hangouts.
We're like 20 feet away from each other.
I was going to go ahead and share my screen so that we can have a conversation around the
Sure.
slides I sent earlier. And I also have all these results, of course, in a 3D viewer, so we could, if we need to pull something up to rotate it or look at it in a little different way, we can.
Okay. I turned off my camera because I think I don't have the bandwidth to.
Okay, yeah, that's yeah, that probably will help speed things up for you. Let me see here.
I'll turn mine on.
Okay. Hopefully you see my PowerPoint presentation.
Yep.
Okay, awesome. I'm going to change the view, so hopefully that's still, is that still okay?
Yeah.
Yep.
Okay, great. So iChen was asking about the preparation of these proteins for docking and kind of what's involved in that. So the structures themselves I got from the PDB from the codes that Paul had sent out in that email, but of course those as is are not suitable for docking because they're missing a lot of information. One, hydrogen atoms are all missing, and we need complete structures to do docking, and also often from the way that structures are determined, there might be missing density for some side chains and loops, so that information is not available it doesn't have any coordinates. So that is not a optimal situation for docking either. So I had to build in any missing loops or missing side chains too, and then evaluate the charges and protonation states of certain amino acids, and make sure that those are all optimal. They're all optimized for docking. So I did this for the receptor, which was that PAD4, and the PAD4 is the biological, or the structure is just one monomer. So, but the biological unit, I just created that using the symmetry mates. And then, as I said, I use the Schrodinger tools to prepare these structures. The main thing with the PAD4 is there was a little bit of a loop missing. I believe it was between, it was in the N-terminus region. I believe it was right in that region where residue 55 is, which I know is an important one for mutations. So there was about 4 or 5 residues in there that were completely missing, that I used Prime, which uses an energy-based calculation to build that loop for that one. So that was important for this. And then there was several residues where the side chains were missing that so we add the optimal rotamer again based on an energy-based method. And then the ligands, if you will, they're really proteins themselves too. I prepared 4 different ones. So we have 4 docking runs, each with one of these ligands, these P65, or a portion of P65, right? So the first one I pulled out was the DNA, the domain with the dimerization domain, and then the DNA domain by itself, and the dimerization domain by itself. And then I-Chen asked if I could also look at another structure, which is, it's a homodimer of the dimerization domain with I kappa beta. How do you, what's the full name of that? I forgot that.
I kappa B.
I kappa B. Because there was, please step in at any time if I'm saying something wrong, but there was evidence in literature that that PAD4 interacted with this via P65. And then again, the same sort of steps were involved for each of these ligands to make sure that the appropriate side chains were all there, and hydrogen atoms. And also because we're now kind of breaking these up into fragments to make sure that the ends of the proteins were treated so that they didn't interfere with docking processes or anything like that. And so then once both the protein, if you will, but both, I say protein, both the receptor, what we're calling receptor, or ligand is prepared, to the strategy I used was to institute a reward for any interaction that the ligand, the PAD6 or the P65 ligands got a reward for interaction with the N term of the PAD4 protein. And then I used again the Schrodinger tool, Bioilluminate, which Piper is the underlying algorithm for that, to do the docking. And then once that docking is done, I started to try to think about the analysis. And it's unfortunate a little bit because I couldn't think of any good controls to do for this, since they're really, I mean, the reason we're looking at a model is because there's no structure of this. So it's kind of anybody's guess as to, you know, what appropriate control, if any. I couldn't think of a good one. But I-Chen said that there were several mutations that are known to affect the activity of PAD4. So looking at those would be a good idea to see if the particular docking pose, how it, you know, where those mutations were with respect to the P65, and structurally, does that make sense that those residues might interfere? And then he mentioned that there is 4 arginines, that undergo cisterlination and affect activity as well. And those 4 arginines are all in the DNA domain.
Yeah, but that's probably not involved in the interaction with PET4 here.
Okay, that
Yeah.
well, you'll see when we get to some of the structures, you'll see that I couldn't see how they're, you know, they don't look like they're specifically interacting.
Yeah, that's go along with our data. Yeah.
So of the 4 docking runs, here's one of the first ones I looked at because I thought it would probably be the most complete, maybe interesting. So this is PAD4, and then the DNA and dimerization domains are docked to PAD4. So PAD4 is shown in green and pink. You have one.
Janet,
Yes.
for some reason the slides aren't advancing.
Oh,
Yeah.
they're not advancing. Okay, maybe it just needs a minute here because of the.
What slide do you think you're on?
So I didn't label the numbers. So it should say at the top PAD4 DNA dimerization domains, and there should be, it's the first structure that we're looking at.
Yeah,
Yeah.
we're not seeing that.
No.
We're 2 behind you, actually.
Oh, 2 behind me. Okay, so you're still looking. Okay, let me change this a little bit and see if it will speed up.
I mean, we could use GoToMeeting if this doesn't work, but now it's moving.
Okay, so this.
Well, but it not. Yeah, there we go.
Here we go. Here we go.
Is this better?
Yeah.
Okay. I think it's because I had it in the presentation mode, which was slow. It's slower.
No, we didn't see it in presentation mode at all.
So we talked about this slide, which
Yeah.
Okay. So then the next slide was just a kind of a continuation. So now we're where we were talking about. Okay, we've prioritized interaction in the N-terminus region of PAD4, and then you did the docking. And then now, you know, to try to talk about some sort of analysis of those results with the different mutations that are known to affect PAD4 activity. So this is the very, hopefully it's advancing now to the very first slide with the structure and the PAD4 DNA dimerization domains. It should be at the top.
Yep.
Okay, great. So this structure here, there's 2 molecules of PAD4. So the green and the pink is PAD4, and here's one molecule. I'm kind of circling that now of PAD4, and then the second molecules, you know, it's basically anti-parallel, and it's over on this side. And I highlighted the pink because that's the N-terminus region where residues 1 through 119, which are thought to be where P65 interacts. And so then the orange you see here is the P65, the DNA domains, that's the DNA domain is here at the top, and the dimerization domain is here. So the DNA domain, it's slightly bigger, and it has this little alpha helix, that's kind of how I keep them straight, is the dimerization domain is just, I have all these beta sheets. So to do this docking the way I did it, I already told you that I gave it I said you're going to get a reward as far as scoring is concerned if you can interact with this pink N-terminus region. That doesn't mean it precludes it from docking elsewhere. It just means there's a bonus if you dock in this spot. So I allowed it to do 70,000 perturbations of the DNA dimerization domain with PAD4 and retain the top 30 poses. And of those top 30 poses, 11 of those 30 poses had some sort of interaction with PAD4. Sometimes it was just a kind of minor, it might have been a loop or something. but in going through those, pose 14 was the first pose to show an interaction with the N-terminus domain, which I'm showing here on the right. So there's an interaction with this in this area here between these DNA domain and the N-term of PAD4. The next pose that showed this interaction was pose 17, and actually it's the exact same pose, but the opposite monomer. There was 4 poses that I felt had like a major surface contact between it was always the DNA binding domain and the N-terminus region. So 2 of them are what you're seeing here, and then there's 2 more that could be explored. They're further down in the list, so they don't have quite as high of a score, but they're, the top 30 I would think would, could all be reasonable given kind of the state of technology for protein-protein docking. So that was the thing, is I never did see in any of these top 30 poses where the dimerization domain was the domain that interfaced with this N-term.
So I have a question here.
Yes.
So I'm looking at the structure, right? You
Yes.
show the DNA domain interact with the N-terminal, right?
Yes, right here.
And then the dimerization domain seems also interact with PEP4, am I right?
That's true. It's just not in the N-term region, and but we can pull this up and see how close. But yes, many times, often is the case. It's interacting with PAD4 just in a different region.
Because our data suggests that both either one, dimerization and DNA domain, can all interact with PEP4.
Right.
But doesn't mean they have to interact with the N-terminal. It could still interact with other part.
And I do see that. In fact, when we go a little bit further on, we can talk about where I just docked just the dimerization domain by itself, so the DNA I was not included, and kind of what happens there too. But before we kind of lose this train of thought, I wanted to kind of zoom in this region right here where you see those interactions. So it's the same structure as before in the previous slide, but now I've just hidden basically all of PAD4 except the N-term domain or N-term residues, and then there's the DNA, the P65. And I apologize if things are not labeled as clear or the colors aren't great. I was putting it together kind of fast. I'm going to zoom in a little bit more here, see if it can. I might lose some resolution. Oops, not that far. There we go. Okay, so here are the 3 residues, 82, 55, and 112, that have mutations, or when they're mutated have affect activity. And yes.
They don't affect the activity of they don't affect enzymatic activity.
Okay.
They're just mutations that are present in patients with rheumatoid arthritis.
Okay, so they're resistance mutation, or are they?
That's a great question. I mean,
we don't really know. Is that the?
Yeah.
Okay. Okay. Well, I wanted to kind of see where these mutations were with respect to where this pose was binding this. I don't know if that's important to you guys or not.
No, it is. I think it's important that because you could make an argument that it all modulates protein-to-protein interactions, and that's how how these mutations promote the development of RA.
Okay. Okay.
So if you get enhanced inflammatory signaling by mutating, then that would, help explain why those mutations showed an increased association with RA.
Okay.
So this is great.
So the other thing on this is just for your kind of structural viewing pleasure, if you will, these arginines, those 4 arginines that Chen was talking about, I've put those in a purple here so we can kind
They're
of.
really all over the place.
They are, but if you go back and then look at it, without the whole pad 4 hidden like I have here, they're not they're like on the other side. Pad 4 is on one side and all these arginines are on, the opposite side. They're not interacting specifically with pad 4, except maybe this one down here by the this 112 residue perhaps. But these are all kind of out there, and at least with this pose in the solvent, what I would say solvent space is.
Well, our data show those 4 arginine are the citronation site, and they should not interfere with the interaction with the PEP4.
Okay, I would say this pose supports that.
Yeah, but another question, I don't, maybe not quite directly related, is that do you think based on structure, are those reasonable citronation substrate site for PEP4 in this structure? Or can you comment on that, or just not enough information to say that?
Yeah, go ahead.
Paul, you have a whole crowd of people outside our office, like 6 people.
Yeah, I know. I'm supposed to be in it.
Okay.
But I'm going to delay it.
Okay.
But just, I'll quickly comment. I'd say most of those arginines are on loops or turns, and those are preferred pad substrates. I think there's one arginine that looks like it's on a beta strand, and that would be a non-preferred substrate.
Okay.
But you never know what that residue looks like in the absence of. Was this structure determined in the presence of DNA?
This was the.
No, there's no DNA. There's no DNA.
But this structure was originally bound. Yeah, I pulled it from a structure that had DNA bound. I deleted the DNA.
So there might be subtle conformational differences with that origin.
Right. Yeah. So let's see. I'm just kind of then moving in a little bit more so you can see where those residues are. This is in that where that alanine 82 and 112 are, and it looks like to me, the 82 said it mutates to a valine, so that of course is going to be a little bit size difference, and it's going to affect the interactions with this proline perhaps, but it's, again, this is a loop region of that DNA domain, so that interaction would change. And also you can there might be some contacts here between this alanine between alanine 112 and 82, so that whole cascade of interactions would be altered by these side chains. And when I see, of course, that the 112 that mutates to a glycine, I also think, well, it's going to change the flexibility, and it's in a beta sheet, so there's some flexibility could be playing a factor of somehow why these mutations are important as well. And then the other residue, the third residue, was this serine 55. It's pretty far away actually from the DNA domain here. These are these residues in the DNA that are closest. Again, it mutates to a glycine, and it's really maybe my thoughts were, of course, it could be flexibility again because it's mutating to a glycine, or perhaps there's something we're missing that water mediated, because there's so much space in here and we're not capturing any waters. The docking, we actually delete all the waters, and there wasn't, if I remember right, there was few, if any, waters in the structure to begin with. So.
Sorry, I got to jump off the call.
Okay.
Thank you, Paul.
Bye. This is great, though.
Yeah. Thanks for joining.
So I have a question. So you comment here that the 82 mutation may increase the van der Waals force, right?
Right, because it's going to be a little bit, I mean, it's more hydrophobic than alanine. And then you have a proline here with its ring kind of, it's pointed in the direction of this amino acid, at least. I mean, you have to keep in mind these are both loops. So, of course, they're going to be subject to a lot of flexibility. But with increasing the size of alanine and hydrophobicity, you're going to have some additional contacts with proline that alanine just doesn't make. So that interaction could be stronger, and that could, that's going to affect this whole cascade of interactions between these three residues.
Okay. So that go along with our data showing this mutation actually increase the interaction.
Okay. Okay. Well, that's good. That's the supports that. And so if the next thing I was going to talk about was the docking with just DNA, with just the DNA domain. If there's no more questions on this part.
No.
Okay. So the docking with just the DNA domain actually is, it's boring but interesting a little bit. So again, I did the same thing with showing that giving a reward if it docked to this N-terminus region for the DNA domain. And 14 of the poses had some interaction with that N-terminus domain. There were several poses that had like some kind of major surface contact between those two regions. And the first pose that showed this interaction was pose 3 and then pose 4. And it's interesting because they actually are the same poses that we see in the previous docking with where the dimerization domain is included. So it's docking it's docking the DNA domain in the same way, which is good, but I think that's a good thing. The one thing that I noticed, I think this is a result of the program, and maybe I need to contact the tech support or project manager for this program, is some of the loops actually show like they're close, like they're clashing or would clash. But I think this might be to a certain extent allowed by the program because of inherent loop flexibility. So I'm still going to look into that. But yes, so you're going to see the same interactions with this particular docking run here. And like I said, there are several other poses that could be explored that were in the top 30. So when we move then to the docking with just the dimerization domain, none of the poses in the top 30 interacted just specifically with the N-terminus region. In fact, but as you were saying, this may or may not be a requirement. There was interaction, and this what you're seeing here on the right is the top docked pose. So here's that dimerization domain, and here's the one molecule of PAD4 on this side. Sorry, that's not really clear where these two monomers are, and then the other side. But almost all of the top 30 poses had some interaction. There's this region that is some beta sheets in PAD4, and there's the accompanying region over here. I will say the dimerization domain seem to very much prefer this region, and almost all of these permutations in the top 30 poses have some interaction with these two regions.
And do you know what those regions are in the PAD4?
Let's see if I have a good picture.
Because it's
right here. It's the domain right next to the N-terminus, so it'd be the next after residue 120.
Okay. And it's kind of different from this location of this dimerization domain in we just missed it.
Oh,
I'm
right.
saying,
Yes, right here.
yeah, this
Yeah.
one,
So wait,
different location.
that's a good point. Like I should check that to see if this orientation showed up. I didn't specifically check that.
So it could be the same region, just different view.
Mhm.
Look at that, right?
Right. Like let me just make a note here for that to check to see if we can find the dimerization domain
Yeah.
in the orientation as the DNA dimerization docking.
So I also have a question on this structure here, right?
Sure.
You have dimer of PET4,
Yes.
but you only have one P65 here, right?
Right.
So can you imagine then there's another P65 there on the other side? In that
Oh,
case,
yeah, like on the opposite side.
yeah, kind of on the opposite side.
Yes, there's
And-
two interfaces.
Yeah,
So you could, yes.
but in that case,
I don't think they'd interfere with each other, no.
but in that case, the dimerization domain won't touch each other, right?
Oh, yeah, no, I see what you're saying there. No, probably not, because they're going to have, I mean, this molecule.
They're gonna
is in
stick
the
to
way.
different opposite direction. Looks
Right.
like.
So that was, I guess, a question I had for you, or even Paul might have been able to answer it too. Is
Yeah.
as far as for docking, I did one dimerization domain, but I was wondering if also because that's the way the structure that I pulled from was, but I was kind of wondering if there should be two dimerization domains here.
Yeah, I think we really don't know whether they interact as a two dimer or two monomer
Okay.
or whatever mixture, right? We really don't know.
Well, I think it'll be, you might find this interesting then. When I went to the I kappa B and with the dimerization domain, that structure has the homodimer of dimerization domain.
Yeah.
And I left it. So we can look at that.
That's the last structure you have.
Yeah, that's the last one I did. So okay, so with this, what that will be a good question. So to see, I'll go back and look and see if we can find that same orientation for the dimerization domain. And then the last one, okay, here is that I kappa beta with the two dimerization domains here, and so it's kind of a big complex. But
Yeah.
so let's see, what I found was 5 of the 30 poses had a slight interact, like this was the, oh, I'd have to go, I didn't write it down which pose number it was. Pose 5, actually, I can, this is, let's see, I'm gonna just make sure I don't, pose 5, shown right. Okay, had some interaction with that end-term region. Again, we can look at other poses if that's not an absolute restriction. But pose 5 was the first one to have some interaction here. And then there was some really interesting, I thought, interfaces that I kappa beta made directly with PAD4 too. Some seemed like some very large surface contacts. In fact, pose 28 I thought was very interesting because it actually, the way it docked, it spanned, the I kappa beta spanned PAD4 to interact with both end-term regions of the two PAD4s. It was quite an interesting pose, I thought. And what I wasn't sure here is, I know you said that there was evidence that that PAD4 interact with I beta kappa via the dimerization domain or via P65. So I wasn't sure if that meant that
No,
that
we
the
don't-
dimerization domain should be the one interacting, and then not necessarily PAD or not necessarily I kappa beta.
No, we have no
track.
information about
Okay.
I kappa B with PET4.
Okay.
Yeah, but I'm just saying one question here is that in this, the last structure, right?
Yes.
You are you are rewarding the dimerization domain with N-terminal PET4, right?
So I gave it a reward for two things in this case, actually. I gave it a reward for interacting with the N-terminus domain and interacting with the dimerization domain.
Yeah, but based on our previous structure,
Mhm.
in your previous structure, dimerization domain actually interacting with the middle part, but not the
Right,
N-terminus.
right, yes. you're
Right. So if you use that one, I don't know what the result gonna be. So say if you
So,
now try to kind of let dimerization domain interact with the middle part of PET4,
Mhm.
would that still fit well? Then we will have the N-terminal of PET4 interacting with the DNA domain.
Mhm. In the
Right.
middle part so with the reward system that I did, what I should do is just go back and look at the results we have
Yeah.
and see if we can again find that same pose that we saw in the DNA dimerization docking, because it doesn't preclude the dimerization domain interacting somewhere else. It just gives it a reward elsewhere.
Yeah.
So
Yeah.
there was, of course, other poses on, and I showed this one because I knew, in kind of my naive understanding, or I should say novice understanding of PAD4, that the N-terminus was important. So that's why I showed this particular pose. But you're right. If that's not a strict requirement, then I should go and look and see if we can find the same pose that we see in that first docking. This one here, maybe with I kappa B. I'm just writing that down. Okay, and then that was kind of it that I had for the slides that I prepared. As I was saying, the thing that was initially stood out to me was that it's not the dimerization domain that seems to be interacting with the N-term, but I think we've gone over that several times now. And then I need to understand, what the criteria is for loop regions in the way it does docking, because some of them seem to be a little close to me. And then, of course, there was a couple other DNA domain interfaces that could be explored other than the one I showed. And then sort of, what else would be next steps?
So one question I had, Janet, about the rewards and the way those work. I mean, are there ways of making the rewards even stronger or more?
Oh, yes, there are.
And having more specific, like attractants, if you like, or tethers.
So you can choose any number of atom or residues that you want as the, like we know, this is, often what we would see this done with like say mutational studies. We know we know maybe 3 or 4 residues are absolutely known to be on that interface, protein-protein interface. So you would use those as your residues to reward. And then it's basically just like a point value system, and you can increase that point value system for rewarding interaction with those specific residues. And I took a default. It was like 0.11, but I think that is completely arbitrary. So you could increase it and kind of see how maybe the the ranking of the poses changes, I suppose, because actually none of these docking cases was the interaction with the N terminus a top pose. So that reward system, maybe my reward system was not enough, or
Right.
Right.
I guess you might start with an extremely strong reward and then back off from it.
Yes. But I think you would only want to do that after hearing what I-Chen was saying with just
Yeah.
the DNA domain specifically, because we don't know where the dimerization domain is going to interact specifically. Is it true, I-Chen, that is it known that the DNA domain interacts with the N terminus region of PAD4, right? Oh, I-Chen, I believe you're muted. Your microphone is muted.
Can you hear me?
I can hear you now.
Okay, because I kind of lost you for a few minutes.
Oh, okay.
Yeah, so we kind of do it separately. So we know, say in in P65, we know DNA domain and dimerization domain all interact with PEP4, but we actually don't know which part interact exactly which part.
Oh, so maybe the reward system isn't good at all.
So we know in the PEP4, the N terminal is probably the strongest interaction part.
Okay.
There's also other part can interact too.
Oh,
Yes.
Is there a paper or some literature that was done to show that it's which residues in the N terminus interact?
No, because this is brand new. No data.
Okay.
We are the first one to show this.
Okay, gotcha.
So you can imagine that you have two domain on P65 and two domain PEP4, and you can do the mix match. We don't know which one interact with which one. Do you see what I'm saying?
Yes, of course.
Yeah. So I have a question for actually Celia and you too. So we talked about this issue before, just that, so this is just all modeling, right? And could we populate this data without any real crystal structure? I think that's eventually, we like to see we can include this information in the paper, right?
I mean, you can do some molecular modeling and think about it as, a potential explanation of the data you see. To do crystal structures is much more extensive amount of work.
Yeah. That'll be the next stage, but I'm saying just by this computer modeling, this data that Janet kind of pulls out, is that good enough in your field? Is that good enough to put into the paper, or you think they're just gonna, people won't take it? I don't know.
People do this kind of modeling all the time. It's not a major, it's more, almost a discussion. It's added to people's discussion of how things might work or what might explain this data, your experimental data.
Okay. So for example, we can show the first crystal structure, the first model that Janet pulled out, and you can say those mutations possibly increase the interaction, and that go along with our functional data. Would that be, you think that makes sense?
I don't, so I don't have your functional data and know how well it corroborates. This still seems like a very preliminary modeling. It depends, to include. In principle, yeah. You can do have a model, but you want it so that it really is explaining your functional data.
It kind of go along with our functional data.
Okay. I'd have to see it. I would have to see the model in the context of your functional data. I can't. It's very hard for me to say whether it's enough to in an abstract manner.
Do you have time if I send you our manuscript and the data? And do you think you can look at that to see it's worthwhile to include Janet's model in there?
what do you think, Janet? It's
Well, I agree with you that I'd probably want to like understand the functional the raw data that iChen has to make sure that it fits. The other thing I would do is I think this would still need a little bit more work before publication, perhaps docking without any rewards at all, and see what the top pose is, since we're not 100% sure where the interaction is. You know, because maybe we're artificially biasing it.
I mean, not knowing your data at all, are you able to, are you,
Sorry.
that's not me, right? Are you able to do, follow-up mutations based on the model, or?
Yeah. So we make those 3 mutations, right? And we found out it interacts stronger with P65. And it also
Okay, but no, from the model that you would predict additional mutations that would test the model if it continues to be reasonable.
Like perhaps in this region or something.
Yeah, are you following?
Well,
So,
yeah, it's just,
I mean, I guess it depends on what stage of your manuscript you're at, and I mean, and where you're sending it, whether or not it would make sense to include a very preliminary model.
yeah. So that's why I'm sitting wondering, maybe if you have time to read it, you probably could have better idea whether it's good idea to include it or not.
Okay, I mean, my time is
I know.
fairly tight, and Janet's is maybe a little bit. I mean, she's been thinking about it. She should probably take first pass, and then
Okay.
I can confer with Janet of whether.
Sure.
But where
Okay.
are you aiming this manuscript for?
Well, I don't know. I think if we can include crystal structure, the data actually pretty good. And I'm thinking we can try immunity first. And it's not working. I'm thinking about natural communications.
Okay.
I think the data is pretty. It kind of all makes sense.
I mean, those are very high level journals. So, I mean, the modeling, so to include a model, I probably would think about, I would think about, having follow-up mutations based on the model to include it in, so, but I don't know if that's practical. You look at the model's based on your data, and then the model allows predictions that aren't there, that you didn't have before. And if you make a mutation and it is consistent with the model, that validates the model and gives your argument even more credence.
We are kind of going from the other side, because this 3 mutation was found in human who had a higher risk of rheumatoid arthritis, right? And then people try to understand why, and our hypothesis that it actually increases interaction with the P65.
Okay.
And our biochemical data fit with that, functional data work, and your models also suggest it could increase the interaction, so that everything seems fit well.
Okay, I mean, yeah, so probably sending it first to,
well, I definitely can send to Janet, and Janet can take a look to see whether that makes sense or not.
Yeah, I mean, I think too, then if we're going to do that, then we need to explore the other possible interfaces of the DNA domain too that came up. I think there was 2 other ones that I just didn't have a chance to look at, ones that didn't score as high.
The thing is that the manuscript we pretty much done. We just are ready to submit it, and I'm waiting to see if we can include this Janet's data.
okay, all right.
My same thinking is that if the data we don't put into the manuscript, then we probably had to wait till we get a real structure to publish this thing. Am I right? Or this could be
Yeah,
published
I
independently?
wouldn't do a model just on its own like this. It would,
Right. No, if it
Yeah.
fits with your data, that makes sense. I mean, you know, to do real crystal, to
It would
really,
take years of work, right?
yes, yeah.
And I don't want Janet to waste time just modeling this thing without getting it published.
Yeah.
So I think it's a decision we have to make whether we're going to continue modeling this. My sense is that you can model it many ways, but could we really use it? That's the question.
Right.
And if we think this modeling is not going to work for this paper, then should we decide when is the endpoint?
Sure.
Sure.
So I think, yeah, I mean, what do you think that's a good strategy? Janet,
yeah, we can, I can take a look at what you have so far in the manuscript. And then, I mean, like Celia was saying, I think that this is a discussion of, this is a probable thing, and that's it, because later it might be proved in a structure that it was wrong, right? So it's not something that could like stand on its own, but maybe within the context of what you're seeing, it might. If I can take a look and read what you have to see if that makes sense.
I think it has to make sense. We also don't want to force it. If say it doesn't make sense, then
Sure, right.
force it, probably going to jeopardize the data. And so I'll send the data and manuscript to Janet to take a look.
Okay.
So another question for Celia is that since that I'm writing a grant and you and Paul thanks for your help. So I try to get money to look at this to make the real crystal structure of this thing. So based on this model, do you think it's worthwhile to generate a real crystal, or you think this just not realistic? I'm totally new to this field, so I don't know what to expect.
I mean, so I don't remember the you know, I don't.
There's crystals of PAD4, and then there's crystals or structures of the dimerization domain with other parts of PAD4, but nothing as a full complex. I mean, as you can see, it's large.
And are you able to express and purify milligrams of these proteins, I-Ching?
Well, I think we can get P65. We probably have to make it truncated. It seems got degraded when you make it. The yield is good, but it just got degraded. And
yeah,
so
I mean, to do crystal trials, we really need to be able to express and purify many milligrams of pure protein. So if you can do that, then that's one, you know, that's a different thing. I mean, the fact that these have been crystallized before suggests that it's possible, but if you can do it in your hands.
Yeah, if we get it funded, I definitely want to purify, make a pure, good protein and for crystal structure.
Right. I mean, the thing about crystallographic grants is if you include crystallography, they basically want to have diffraction in hand. Right? They don't, you know, saying that I might do crystallography one day usually doesn't fly.
Yeah, so this is just part of it, and I think the first round review, it was not funded, but actually they do not have negative comment about this crystal work.
Okay.
They just want me to describe it more in detail, which I have trouble. I try my best, but they don't have negative comment about the crystal work, though.
Well, that's good. I mean, so I mean, probably sending the reviews. You know, I don't think I don't know if I saw. Did you send the final grant? when it got submitted?
Yeah, we sent it last time, and it got a score, but not in the.
No, no.
in
Did
the-
you send it to me? Do I have a copy of the grant?
Oh, I think I probably not. I think may send it to Paul. I don't know. I can send to you again before I submit it this time in November. Yeah.
Yeah.
And,
I mean,
I'll appreciate that you can make comment on those description of crystal work. I can
sure. I mean,
copy
you know,
copy from your paper.
okay.
I don't know what I'm writing. I just copy some of those kind of material.
Yeah, I mean, it depends how extensively and how, I don't remember offhand how much you budgeted for us to do. I mean, it's a lot of work. So it's basically a full-time postdoc to really do this.
Yeah.
And supplies and things. So, I mean, and that's, that's real money. I mean, so if we're serious about going for that, that's, that.
I think we have a subcontract, right? And I think we are working on it right now. And I think Paul, I always CC you and Paul together.
I know. I'm often very busy with.
I know.
I'm juggling many things. I mean, I know Paul is too, but it's systems he's more familiar with than I am.
Yeah.
But I'm back. But
okay.
It would be useful to share the grant and to chime in. And I agree with you that Celia, that it is a lot of work to be able to do this. And if we're going to do it, we have to do it. Yeah.
You have to do it right if we're doing it, and think it through a little bit. Yeah. So I mean, what was the score on the first submission?
I think he got scored 20 percentile.
Oh, 20 percentile. So it was very good.
Not too bad. Yeah.
Oh, that's very good for a first submission.
What study section each in?
I think
Or what institute?
I think there's autoimmune hypersensitivity. I think HAI study section.
Okay.
Yeah.
But we can express milligram amounts of PAD4, and if we just did the immunoglobulin, the N-terminal domain, the first immunoglobulin-like domain, or even the first two, I would suspect that we'd be able to express larger amounts, which might make it easier to crystallize, because then you don't have to deal with the catalytic domain.
Yeah, I think Paul, last time we talked about making the P65 recombinant protein, and we keep getting a degraded product.
Yeah.
And I think you were talking about try to maybe cut the full length one and try to purify in some way. But I think that we haven't done anything on that side. Just waiting to be funded. Yeah.
I mean, so just remind me, so because I've been in and out of the, because I'm not quite up to speed on this yet, but you think that it's the IG1 domain of PAD4 that interacts with, is it the DNA domain of P65?
Well, as I say, we don't know. We think there are two domains on each protein that can interact. There are two DNA domain and dimerization domain on P65, and then the N-terminal domain and also middle part on PAD4, right? But we don't know exactly which one interact with which one. You can see two domain on one protein, two domain on one protein, and you can do mix match. We don't know which part interact which part.
Yeah, if anything, I would do, you try, I mean, it looks like you can get the P65. There must be crystallization, or there's crystallization conditions, so there must be expression conditions for that domain. So we could start with that domain and then try to co-crystallize with full-length PAD4 and then the IG1 domain and then the IG1 and IG2 domain. We can't do middle stuff because we've never been able to express truncation mutants of PAD4 in which we started, in the middle of, or where we sort of chopped off the IG1 and IG2 domain and tried to just express the catalytic domain. because for whatever reason it just goes into inclusion bodies, at least in criteria. So, in my mind, the sort of simplest starting point would be to take the known expression conditions for the DNA and dimerization domain of P65, and then, as I said, try that co-crystallization with full-length PAD4, and then the
Yeah.
IG1, IG2, and then IG1.
Yeah, that's my plan too. Okay, so I think we try to set up the subcontractor with you two. So I think you may want to review the subcontract, make sure that we have enough money to do the work, and we can submit it. Hopefully, they'll fund it. Okay.
Celia's on mute. There you go.
Yeah, I mean, I think that makes sense. It would be good to actually see the grant and see how much, what all you're trying to do. I mean, if it's, and
Yeah.
talk about who's going to do, if the division of labor a little bit. Is it? I mean, when you submitted it, we were on it, but I didn't really follow the details of what we're hoping us to do. So
I know you're very busy, but yeah. So yeah, I will send you that part of the grant, and you can look at that to make sure it makes sense.
I mean, can you send the whole grant so you understand how it fits in the bigger picture, or are you not comfortable
Sure.
doing that?
I can also send you the entire. It's just still in the draft stage, probably not complete.
No,
yet.
but the previous submission,
Oh yeah.
the comments, so just can have a sense of, and maybe just highlight the region and what.
Okay, I'll do that. And I will send the manuscript and figure to you, and Janet can take a first look to see that would that make sense to include those your modeling in there.
Yeah,
and
on the paper, yeah.
And if that's my sense, if it's not in this paper, then I'm not sure we want to keep doing the modeling things. I don't know. I'm not in this field. Maybe you can have better idea how to pursue it. Yeah.
Yeah. I'll look over the manuscript.
Okay.
I-Ching, just to confirm, you showed in the manuscript that the three mutations interact more tightly with.
Yes. Yeah. And that's same as your data in that paper, right? You show it interacts stronger with other protein.
Yeah.
Right. And so I'm just saying that the model seems fit well with our data, and that seems will merge very well with our paper, and that will probably make it better. But I'm not a crystal person, so I don't know what people will they accept the modeling as a way of data, or they just think this.
I mean, people publish molecular models all the time.
Okay. I didn't know that.
Especially if it's just it's basically part of the discussion. It's often used, as I said earlier, as part of the discussion piece of how to potentially explain the data. It's not sold as proof that this is the way it is, but this seems to be consistent with what we've observed.
Okay, that sounds good.
I mean that. Janet, that's a reasonable way.
No, that's exactly what I was saying. Well, I-Chen and I were talking about last time. Is it's kind of centered around this is a possible mechanism, may or may not be right, but within this context it makes sense.
But because it enhances interactions, you have a couple of things would increase the van der Waals contacts with P168. So you could it offers some explanations.
Right.
Whether it's true or not, I mean you have to add caveats. until you have a real structure of the
Yeah.
complex, you'll never know for sure.
Mhm.
Right.
So
no,
Paul, you read the manuscript, right? And
Yes.
so you think that will fit? With this Janus data will work well together with our data?
I think you could, I think it could be discussed, and as Celia suggested in the discussion, and whether it's a supplemental figure or a main text figure. I mean, that's something to think about. But I think we definitely could include it as a possible explanation or
I mean,
rationale for the interaction.
what I was saying when I think you weren't online, Paul, is that I mean, I don't know what stage. It sounds like the manuscript says is at a much different stage along the line, but that what models are often used in both to validate the model and to further explain the situation is that you use them predictively to engineer mutations.
Mm-hmm, mm-hmm.
Yeah. And then that further validates the model and that you're barking up the right tree, basically.
Yeah. No, I agree with you, Celia. Although you could slightly spin it that you did this modeling, and it suggested that it would improve the interaction, and then you did the experiment because he does have data showing that when you make the three mutations,
Mm-hmm.
and they're called the SNPs, the RA associated SNPs. When you make those, that you would predict that it would enhance interactions, and then you would test that hypothesis. So I mean, you could frame it. I think you could. I guess I think in the paper you could frame.
I think we can frame that way. Yeah.
Earlier, as suggesting that the mutations might enhance the interactions, and that's why you then tested the SNPs, the RA-associated SNPs, for an effect on binding to P65.
Yeah, that's what that's my thought too. Yeah. All right. Well, great. Thank you very much for your help, Janet, Celia, and Paul. And I'll send you the manuscript.
Okay, that sounds
And
great.
I'll send Paul and Celia the grant, and you can look at that.
Sure, that would be good. Thanks.
Yeah. Okay.
Okay.
All right. Have a good weekend.
Thank you very much. Have a nice week.
Thanks.
Okay. Bye, Ashley.
Bye.
Bye-bye.
Thanks a lot.
Thanks, Janet.
Yes, no problem.
Thanks, Celia.
Yep.